All right, chapter 12, the cell cycle. It feels really strange to me to be giving this lecture right at the end of the semester, right? Normally this would be middle of the semester and we'd be kind of moving into genetics. And so at this point, we've already talked about meiosis, which is kind of the cousin of mitosis. And so a lot of this should be review, and that feels a little strange right now, but that's okay. A good time to review some of that genetic information, things like DNA replication, and then we'll talk about the cell cycle, and we'll talk about cell division, which is mitosis. So, and I can't believe this is our last chapter. Nice job on making it this far. I know this hasn't been the easiest semester ever, so hang in there. Almost there. So, there are also, I will say, in this chapter, some phenomenal images where they've actually captured different parts of the cell by different staining techniques using different fluorescent markers on different aspects of the cell. And so this, I think, is a really nice example here of cells undergoing division. They're showing division again there. Seeing the actual protein fibers, the microtubules, the chromosomes are stained in that blue there. Anyway, again, here's another one kind of close up. So in red, we can actually see the kinetic or the microtubules, the proteins inside of the cell, and we see them reaching out and attaching at the centromere, at the waist of the chromosomes. And this would actually be in metaphase. We're getting ready for this cell to divide. Again, lots of pictures of cells undergoing division. So a couple things to keep in mind here, right? Depending on what organism we're looking at, this cell division might have very different purposes. So up in this top picture, we are showing that in a unicellular organism, when a cell divides, it's actually reproducing the entire organism, right? So that would be asexual reproduction, two brand new organisms. Now, in multicellular organisms, multicellular eukaryotes in particular, we, like humans, we use cell division for a couple things. One, development from a fertilized egg, right? So when a sperm and an egg come together and form a zygote, you have a single cell. And in the 9 months of gestation, you're going to need to divide and divide and divide, so that by the time the baby is born, it's got like millions. Man, how many cells? Millions of cells, right? It's had to undergo division a lot, right? So that's going to be important in growth and development. And then we also use mitosis, we use cell division in order to repair or replace or renew, as they're saying, tissues. So, you fall off your bike and take all the skin off of your arm, you have to replace it, right? Cells that are getting old and worn out, like your red blood cells, right? They only last for 120 days, and then we're going to replace them. Okay? And so this is a really important role of cell division. I will point out that cell division is really a small part. It's an integral part, but it is a small part of what we call the cell cycle, the life of a cell from when it forms, right, from when the parent cell divided, right, so when it first forms a cell to the time when it divides. That's considered the cell cycle. Think about that. In a human, we would talk about the life cycle, right? So from when you're born to when you die. Now we're talking about cell cycle. Okay, so again, a little bit of review here. So in this eukaryotic cell, what we're looking at are the chromosomes. And remember, in humans, we have 46 chromosomes, right? 23 pair. And each of those chromosomes is made out of DNA wrapped around proteins, right? The DNA in the cell, if you take all this DNA on all these chromosomes, that is the genome, right? All the genetic material. And one thing to keep in mind too is that we actually, chromosomes are only really visible when cells are actually getting ready or undergoing division. Okay, so this is a scanning electron, use of a scanning electron microscope to actually get this image of a chromosome, right? Because think about how tiny this is. So this is a chromosome in its condensed form. It's all coiled up. It's ready to undergo division. And also notice it's at that point where it's already duplicated. So remember, see, we've already done this. Remember our sister chromatids? Right? The sister chromatids, basically one of these was a chromosome. It did DNA replication, and now it has a sister. And we can go more into that because maybe this is a thing that we missed last time, and maybe it'll make sense this go around. Okay, what else about this chromosome? Oh, and this is, I mentioned that waist. This is the centromere, see, with an M. So one thing that people get confused when we talk about mitosis is there's a centromere and a centrosome. The centromere is where the two sister chromatids are actually held together. Okay, so again, this confuses people, right? A daughter cell at the end of mitosis is going to have, right, the 23 pair of chromosomes. They're just showing one here, right? But it just looks like this. And we're going to see that the cell, before it can actually divide, it needs to duplicate that chromosome, right? So we do DNA replication. Remember, helicase comes in, unzips the DNA. We had all the polymerases, right? And so we take this one strand of DNA, we make two, right? And now we have sister chromatids like we saw in that previous picture. And then what we're going to look at is right here is in mitosis, right? How do we pull those sister chromatids apart? So again, people get really excited about mitosis. It's like the sexy part of the cell cycle. There's a lot of stuff happening. We can image the chromosomes, and it's pretty cool. But realize, sorry, I'm trying to figure out the pointer. Realize that it's actually a very small portion of the cell's life cycle, right? So when we look at the cell cycle, so this new cell, when it's formed, it's going to spend the majority of its life in what's called interphase, right? Kind of like the in-between phase. And that's like 90% of the cell's life, right? Or the cell cycle. And it spends time in these three different phases. Gap 1, S stands for synthesis, right? This is when we're going to actually copy that DNA or duplicate the DNA, and then gap 2. And we'll look at a couple important things that happen in here, but realize this whole time in interphase, the cell is being a cell, right? So I tell my students, it's like, if, I don't know, maybe you're not old enough, maybe your parents aren't giving you a hard time about not having kids, right? Like procreating, I'm comparing that to mitosis here, right? Like procreating is the only important thing you could ever possibly do with your life. And you're like, but mom, dad, like look at all these other things, right? Like that are going on in my life. So I encourage students to think about that in a parallel here with the cell, right? It's in interphase, it's living its cell life, right? If it's a liver cell, it's detoxifying alcohol, and it's producing clotting factors, and it's housing all these macrophages that are help keeping you free from illness. I mean, right, the cell is doing the cell things. If it's a cell in the pancreas, maybe it's producing insulin. That's really important, right? Life is not all about mitosis, but mitosis is really cool. And I think part of what goes on is, where can I get out of the way here? I think part of what's going on is this is when you can actually see the cell doing something, right? This is when the chromosomes are visible. The rest of the time, the cell's just being the cell. So what we're going to see, and we're going to work with this in lab this week. So this is, if you can get a hold of this language now, it's going to help you complete lab this week. So again, here's interphase. Right, so cell just being a cell. Notice if they're saying this is in gap 2, G2, so the chromosome has already duplicated, right? We're past that S phase, but notice how it's just all throughout the nucleus. And this is what I was saying, chromosomes aren't really visible. The DNA doesn't have to like coil up as tightly until it's getting ready to divide. So the DNA, the chromosomes are just kind of hanging out in the nucleus. They're pointing out that the nuclear envelope is complete. There's the nucleolus. Remember, that's where ribosomes get built, all that kind of thing. So then we enter prophase. a couple key things that happen here in prophase, right, is that we see the actual chromosomes start to condense, right? They're supercoiling up and they're going to start looking like those linear chromosomes that we're used to. We also start to see, here's the centrosome, right here. Remember I told you centromere and centrosome confuse people. So the centrosome has what are called centrioles. It has two centrioles, or centriole pairs, and these are going to be what builds the microtubules, what builds the apparatus that's actually going to rip our genetic material in half. And so if you see them talking about the mitotic spindle, it's part of mitosis, and it's going to be all these protein fibers that are reaching out for the chromosomes. Okay, so in prophase, we start to see the DNA condense, and we start to see that mitotic spindle developing. In lab, we will not use prometaphase. It's really hard under the microscope that we're using to distinguish this from other phases. But notice a couple things that happen here. So importantly, the nuclear envelope fragments. And so notice now the DNA is kind of spreading out more widely, right? It's not bound by that nuclear envelope anymore. The other thing that we see here, right, is that those centrioles have moved to opposite poles. Okay, so they're on far opposite sides of the cell. Again, you won't be able to see the centrioles in lab, but we'll end up grouping prophase and prometaphase together for our purposes in lab. But again, the nuclear envelope fragments, and they're pointing out, they're using this as a time to describe some of this terminology about the microtubules, these proteins. So there's kinetochore microtubules, and notice these are the ones that are actually attaching to the waist, the centromere of the chromosome. And on that waist, there's actually these special protein patches called kinetochores. And so these microtubules reach out and they attach on that patch. And then there's also non-kinetochore microtubules. And these are basically just these proteins are growing, building towards the opposite pole of the cell. And then they're actually going to like push past each other to help rip the cell apart. Okay. It's this image out of your book, 12:7, actually packs a lot of this information in. If you're not excited about reading the paragraph, a lot of the information is actually in here. Okay, so then what's going to happen is those kinetochore microtubules, when they've reached in, there should be one from either side that's attached to a sister chromatid, right? So if you think about that chromosome with the two sisters, we're now reaching in from either end and grabbing one side or the other. And so those kinetochore microtubules, they go through this tug of war until they pull all the chromosomes into this band across the center of the cell. And this is actually like three-dimensional, right? They're not just like in a line, but they're covering kind of the whole center of the cell. And so they call that the metaphase plate. This is the stage of mitosis that's actually super easy to recognize because all of the chromosomes are right along the middle, metaphase middle, okay? And then what's going to happen is our game of tug of war continues, and as we go into anaphase, we actually manage to pop those sister chromatids apart, right? And now each individual sister is just called a chromosome again, right? They had the exact same genetic material, right? We just had made two copies knowing that we were going to undergo this mitotic event, this splitting of the cell. And so in anaphase, and it literally is, right? Picture this protein just yanking this chromosome down towards this end of the cell. And so you can kind of see his like little arms or his little legs are left pointing towards the center of the cell. And so you can actually see that in anaphase. In the final step of mitosis then is telophase, and we're basically getting two new nuclei, right? So we've completely divided the DNA. So you start to see them stretching out at either end. Cytokinesis is really the pinching of the cytoplasm. So you start seeing this cleavage furrow, this indent here. Mitosis is really nuclear division. So that ends at telophase, but then cytokinesis allows us to split the cell. And these are apparently, oh, I left these in. All the same pictures, just larger. You have them in your book, so you can go back and look that way. But I just thought these were phenomenal images. There's Metaphase. Again, it's not like a clean line like they showed necessarily in the cartoon, but as you start to practice and look at these, it does become pretty obvious. Anaphase, they're getting pulled away. Telophase, right there at opposite ends. Okay. And so again, really what's running this show are these structures called the mitotic spindle. And this is made out of microtubules, right? So this would be part of the, like a cytoskeleton, right? And they control the movement of the chromosomes. Again, it's the centrosome where we organize these two centrioles that are putting out the mitotic spindle. Again, they need to be at opposite ends of the cell in order to start this tug of war. Okay, I think I went through all of that. So one thing, oh, this is a nice picture. So notice in this picture, when you're looking at the chromosomes, they're actually showing almost like a little black button where that microtubule attaches, and they are trying to show there's this little like protein patch called a kinetochore, and that's where that microtubule attaches to. Now things work a little bit differently in plants and animals. So again, cytokinesis, I pointed out that cleavage furrow, right, as we start kind of getting ready to pinch the two cells apart. But notice in a plant cells have cell walls. And so this is actually going to be, they build what's called this cell plate. It's going to turn into this new cell wall. And so that's visible. And so that can be helpful for us because we're actually going to be looking in lab. Sorry, keep being in the way here. We are going to be looking at mitosis taking place in plant cells. And so that can actually help us make some of these distinguishing, when that cell plate, the new cell wall, comes in, that can actually help us distinguish what phases we're in. So these are really nice pictures. Again, we're going to be working with onions. We're looking at plant cell walls. So notice here, like we could easily call this prophase because we're actually seeing, I wish I had an interphase one. This is interphase right here, right next to it. Just kind of blah. Here in prophase, right, we should actually be able to start seeing the chromosomes condensed. We're going to, again, group prometaphase and prophase together. So if you see either of these, like, hey, those chromosomes are condensing, we'll just call it prophase. Metaphase, see how they're, again, it's not a perfect line, but the wastes, the centromeres, are all right down the center of the cell. Anaphase, they're getting pulled away, and then telophase, they're starting to form two new nuclei, and you can, in this case, actually see that cell plate. So we're going to be going through and counting cells in different phases of telophase as we do a little experiment. Okay, speaking of which, make sure you answer those 5 questions on the lab before you come to lab. Get you thinking about this ahead of time. So one other form of cell division, so we just noticed the difference is between plants and animals doing cell division. When bacteria do cell division, it is not technically mitosis, right? They do what's called binary fission, and this is their reproduction. This is asexual reproduction. And really, the big distinguishing thing that I want to say, right? So some things are really similar. So for example, before they can divide, they have to duplicate their chromosome. But remember, bacteria have a single circular chromosome. So you run those origins of replication, both directions, right? And you can flip that cell open, or the chromosome open, and you have 2 copies, right? So they're pointing out here 2 copies. But what we're going to see then is that they kind of move to opposite ends of the cell, and then the cell pinches off. Notice there was not a mitotic spindle, right? There's not all these proteins, there's no tug of war, because it's only a single circular chromosome. So it's a little less visually dramatic, but they can do this really quickly, right? If you think about how fast bacteria can multiply and grow and thrive. Okay, so then the last real thing that I think is super important in this chapter, they do talk about checkpoints in the cell cycle. This is super important in understanding cancer. I mean, it's important in understanding how cells know when to divide and how long their life cycle should be and things like that. But really where I tend to see the application of this is in something like cancer, where cells are choosing to ignore these checkpoints, right? And so all they're trying to show you here is that as the cell is moving through growth 1, right, it hits this little wall. And then we get to decide, okay, is that little flap going to go down? And we get to continue moving through here, right? They're showing like this little bar. Do we get to keep moving through here, or does one of these checkpoints stop us? And these checkpoints are specifically placed in phases where if there is something wrong with the cell, we can make it stop. And so, for example, this G1 checkpoint, if something seems to be wrong with the cell, like a mutation, something's going on with the DNA, that cell will actually just like circle out and enter what's called G0. So then just picture it like, oh, you're never going to divide, but you can sit over here and do your job. And so that's what G0 would be. But again, this isn't all about cancer. Some of it is about like how frequently cell division should take place, right? So we know like our skin is constantly sloughing, and so we need to keep cranking out new cells, new cells. Our liver, those cells like barely divide. And so different cells are going to run this at different rates. Okay. So one of the ways this is done, right? So like how skin and liver cells know to replicate at different rates. This gets a little complicated, but just hang with me here. Look at this. So there's this molecule, it's called a cyclin-dependent kinase. Actually, this is the one I wanted to look at, cyclin, right? So this is this molecule. And notice like in the S phase and into G2, it's accumulating. It's building up as the cell is getting older. Right? So this is a way we can kind of time how mature or how old our cell is. And what we'll see, this gets kind of complicated, but there's this kinase, so that's an enzyme, right? These two are going to fuse together into what's called this maturation promoting factor, right? And that is what's actually going to kind of open that gate, that checkpoint at G2 and say, yes, you should go into mitosis, you should go into cell division. And notice then in this process, the cyclin gets degraded, the enzymes don't get used up in a reaction. So it just keeps hanging out. And then as that cell gets older, more cyclin accumulates. Okay, so again, just think of this as a way that the cell can kind of tell how old it is and understand when it should be ready to undergo mitosis. Okay, so there is a lot going on in this picture, right? But here we're showing, oh, here, this is nice. Get out of the way. Okay, so here's that G1 checkpoint, and they're just showing you, hey, there's two options here. And this is what I was talking about, that G0. So if a cell is in that gap 1 and approaches this checkpoint and it's like, something's wrong, you go into G0. You go into timeout. But if everything looks good, you get the green light, that gate opens, right, and you keep moving. The G2 checkpoint, right, same idea, is our cells going to be allowed to enter metaphase? And we were just looking at how that one is opened by those cyclin-dependent kinases. And then the M checkpoint, right? So this is in the middle of mitosis. And that one is basically a chance to check, okay, did mitosis occur properly? Does it look like we actually divided the cell material appropriately? Too much talking. Okay, so these checkpoints then are regulating the cell cycle. And these become really important, how do I say this? In cell behavior. I should say too, because I'm talking about things like those cyclin-dependent kinases, like that's all internal, right? As the cell gets older and accumulates that protein. But there's also kind of external factors that influence cell division as well. So different, maybe different like growth factors. So there's one called like insulin-derived growth factor, or you'll come across one called VEGF, vascular endothelial growth factor. These outside chemicals that kind of work in here and help regulate these checkpoints, right, depending on the needs of the tissues in the area. But as these influence the behavior of cells, what we'll see is that normal cells and cancer cells behave really differently. So this one, they're pointing out what's called anchorage dependence. So normally cells that break free from their tissue are going to undergo apoptosis, right? They actually need to be attached to a surface. They need to be with their neighbors to want to undergo cell division. Normal cells exhibit this density-dependent inhibition. So here's another one of those, like, okay, we're getting signals from the cells around us that kind of check our growth. So if the whole bottom of this tray is filled with cells, they stop dividing, right? They're getting that signal. The checkpoints are kind of holding them, okay? And so here they're just trying to show, okay, if we remove a few of the cells nearby will divide and fill that gap, right? That's how we actually repair our tissues, which is so crazy. But what we're going to see is when we're looking at cancer cells ignore these normal cell signals. So they don't need growth factors to grow and divide, right? To run those checkpoints. They make their own growth factors. So they can send signals that they should keep dividing even when they've filled a space, right? So they don't exhibit that same density-dependent inhibition. They don't stop. They just keep growing. They also, if they break free, right, we call that metastasis. So they're trying to show here's the cancer, some of it's going to break free, maybe moving into the cardiovascular system or the lymphatic system, and those cells don't undergo apoptosis like normal cells would. So what we'll find is, right, so thinking about cancer, if those abnormal cells are still at the original site, it's called a benign tumor, right? We typically don't call it cancer until we're talking about a malignant tumor, one that's actually invading surrounding tissues, one that can undergo metastasis, one that can break free and move to other parts of the body. Okay. So one last thing, sorry, I could talk about cancer for a while there. It's really fascinating and some really amazing new research that's going on where it really feels like we're getting the hang of understanding really the behavior of a lot of cancer cells. Though I'll also say I really think we should but we really should spend more time figuring out why cancer rates have risen so much. Do we really think it's a good idea to surround ourselves with so many chemicals, right? Does everything need a flame retardant? Does everything need a pesticide? Anyway, okay. So the last thing I would encourage you to do then, I included this image because in lab, this is what we're going to be doing. We're going to be looking at cells near the tip of an onion root, and we're going to be recording what phase of the cell cycle that cell is in, right? And some of these are super obvious, right? This one right here, I would say is undergoing anaphase. Ooh, look at telophase right here, right? You can start to see the little cell plate moving through here, right? There's a nice prophase, right? So these would be interphase. Interphase, right? Don't get confused. Those two black dots, those must be nucleoli. Where's the metaphase? I might not have a nice metaphase on here, right? But anyway, this is what we're going to be doing. So maybe spend a little time looking at that, seeing if you can get your identification skills down, and we'll look at the effects of some chemicals on the rate of mitosis in onion root. And you do have to make your own slides. We actually get to do a couple cool things in this lab. You'll have to make your own slides and then identify what phases of the cell cycle they're in. And when I say the cell cycle, don't forget interphase, right? Remember, that's where a cell spends 90% of its life. Okay, we will see you guys in lab. Have a good one.